in, Shav.
Okay.
Glad to hear it.
How many people are they usually having show up?
Recently it's only been a couple people showing up. You and David mostly. I'm glad to see Shivani's back, and Karen, we're glad to have you. Feel free to use your microphones if you'd like. Makes it easy. And we'll give it another 2 minutes to let more stragglers come in. And then I wanted to play Jeopardy as kind of a test review this time. The only problem is it won't let me click randomly which questions, so we'll just kind of have to go down the row question by question. Hope that's alright with everybody. Shivani, it looks to me like your microphone should be on. Karen's is now muted. If it's not turned on, you can go up along the top. Yeah, didn't hear you, Shivani, so maybe go towards the top where it has the picture of the microphone and see if you can enable it there. And also, if microphones aren't working, darn, yeah, still not working. So, if microphones aren't working, another option I was thinking of, since it's similar to a quiz, there's an option where you can raise your hand on the left screen, and we could just kind of rotate through people answering that way as well, so everybody gets a chance. What are all of your thoughts?
I don't care.
Okay. Let's try it, see how it goes. So, welcome again, everybody. Oh, darn, we just lost Karen. Hopefully she'll come back. These questions are pretty much straight off of Dr. Rosen's study guide questions. So they're all things that she would like you to know for the test, and we can kind of expound as we go. So, Darwin for 1 point. What were the key observations that helped Darwin develop his theory of evolution?
He's been developing his theory? What he did, or
there was a slide on week 10's lecture that had three things. Yeah, there were 3 things that Darwin noticed that kind of sparked his curiosity of evolution.
Oh, Darwin's main 2 points Origin of Species.
That's the
I'm
book.
looking at this slide right now.
I think it's a slide right before or right after that. There were 3 things, A, B, and C.
I've got 4 on one and 4 on the other. Then it says the origin species Darwin proposed a hypothesis, a scientific explanation for his observation. He used hundreds of pages in a book to describe the evidence supporting his hypothesis, made testable predictions, and reported the results of numerous experiments he had performed. Is that what you're looking for?
No. It's on slide. I'm pulling up the slides right now.
No?
One of them had to do with insects and environment. Do you remember that slide?
Natural selection? What number is it?
I'm looking right now. Let's see. My computer isn't wanting to pull up both at the same time.
I got one that says observation, overproduction, and competition, individual variation, exclusion, unequal reproductive success.
It's the one that talks about the diversity of life, the patterns of similarities in an insect suited to its environment. Why is it not wanting to pull up? Here we go. It's on slide number 9 of week 10.
Like patterns and similarities. It's like that insect within their environment.
Yeah, so why do we think each of these helped Darwin to start thinking about evolution? How do each of these relate, I guess? Yes, Shivani. Oh.
I think she's probably writing it.
I think so, yeah. The patterns of similarities suggest that we have common traits. Exactly. There's a common tie. Exactly. So if we're all related in one way or another, then that sparked Darwin into thinking, okay, well, if we're similar in some ways, but yet we're diverse, exactly, there had to be change at some point, right? And when was that and why? These are all kind of questions that Darwin started thinking about. How about the third one? I bet you're writing it right now. Let's see. Change came about because when we had to adapt to whichever environment we lived in. Exactly. There are certain characteristics that are better suited to different environments. And so we have to be diverse and change in order to meet that. Alright, so what about this question? Why do flies have so many offspring?
Because a lot of them don't live. Not so cockroaches. If two cockroaches have all their descendants survive after 7 months, there's 1.64 billion of them. So,
Exactly. And why don't they all survive? Do you have any ideas, Shivani?
If you look at
Oh, no worries. Go ahead.
there's 500,000 spores every at the peak of production, some fungi can produce 500,000 spores per minute. Each spore is capable of growing into fungi.
Exactly.
They all grew. They all grew. There's no way that. But with natural selection, some die off, some don't, depending on what happens in the environment.
Yeah. And also they're going to compete with each other, right? The parents only have enough resources. Oh, sorry, go ahead. Okay. The parents end up having a whole lot of offspring, right? But they only have enough time and energy and resources to care for some of them. So the offspring are going to compete with each other to see who can survive and who can be the most fit, I guess. So even though they have a bunch of offspring, only a few end up surviving. How does natural selection work?
An advantage to them. Typically, individuals with the best offspring and passing it off. That is on slide number 20, actual flush. I don't know if that.
Yeah, Brandon, your mic's going in and out. Can you say that one more time?
I did what is on one of the slides, and it said within a population, some individuals have variations that confer an advantage to them. These will be the individuals with the best odds of surviving and passing their genes to their offspring.
Awesome. So, in your own words, what does that mean?
There's variations that we have, and that will pass on to our children in generations beyond, and the ones that are and the better off. I don't know exactly how to say it.
You have it. The ones that are better off
are the individuals that are our children will have better odds or better genes than we did, and they're because of the genes that we passed on.
Exactly.
Yeah.
So the better genes. Yeah, that made perfect sense. So the better genes we have, the greater our chance for survival, right? And those who do survive are able to pass those good genes on to the next generation. They're able to have children who also have good genes and are more likely to survive. And the example of that in the book, or in the slides, exactly, Shivani. Those that come up with a way to survive, those that adapt, are going to be able to reproduce and pass it on so their children have more chance. The example in the slides was the beetles, with either black, gray, or white. Yeah, what's up, Shivani? Yeah, there's a question.
Yeah, there's a question.
Could you explain the environment selecting for and against? Okay. Yes. So, oh no, both of us are now? Hopefully, this will work better. So, let's think of a trait. So, I'll use the example I used in SI last week, I believe it was. People who have darker skin are more able to do well in sunny, hot climates because the pigments in their skin provide a protection against UV. So the environment, meaning the places close to the equator where there's lots of sun, those environments select or prefer people who, so the environment, the sun, selects dark skin and selects against lighter skin. Exactly. In a different example, let's see. If you think of the environment selects the ones that survives, basically. Yes. So the environment selects the better traits. And then if it selects against something, it means that somebody is less fit, less likely to survive. So we'll keep going. Whoa.
Yeah.
Here we go. Next slide. So the front leg of a cat and the wing of a bat both have the same bones in the same order. This is an example of an... Oop, Shivani's got it. Hand up fast on that one. Homologous structure. Exactly. And just a reminder, the prefix homo means same. So same structure in both cats and bats. Awesome. That was good timing. Alright, next one. An inherited characteristic that improves the odds of survival is called an... Bueller. I couldn't help it. So how do you improve your odds of survival? Genes? Okay, what do you do to the genes? Not sure, okay. Any guesses from you, Brandon?
I'm looking, I can't find it. I don't know. I'm looking for it, but I don't know where.
I think it's a slide with a dolphin on it, I believe.
Oh, that's before it. Okay. Adaptation. An inherited characteristic that moves the odds of survival and enhances reproduction. Diversity of life at every level from the gene to adapt.
Exactly. If you want to improve your chances of survival, you have to adapt to the environment. Make changes so that you can better fit your environment. Any questions with adaptation before we move on? Okay. Oh, too far. Alright. What?
Nothing. Never mind.
Okay, I hope y'all didn't see the answer to that because I totally clicked too far. But now we're into biotechnology. So what type of cell is used to provide the nucleus needed in cloning?
Is that lecture 9?
That is lecture 9.
No, somatic. Totally read that on the other one. But it's
So you had it right, somatic. So what is a somatic cell? What's the difference between it and other cells?
it's from a female, I don't know if that's correct.
It's any cell that's not a gamete. The reasoning for that is because it needs to be diploid. Because when you're cloning, you don't fertilize the cell, correct? You just put in an already diploid, already full gened nucleus from a somatic cell. And that somatic cell... Oh, repeat the question? Yeah. So, what type of cell is used to provide the nucleus? We use somatic cells because it's diploid. We don't fertilize a cell that we're cloning. I think that's the question I asked. I forgot, Shivani. Sorry. So that's it? Alright, good. So yeah, we need all of the genes, and you can get that anywhere except the gamete. So what vector was used to get human insulin gene into a bacterium? I've asked this one before in an SI session, I think, if we got to it.
What slide number is it?
That's what I was just looking up. It's on slide number... It's on slide 48 and 49 in lecture 9. Not sure what I mean by vector. Okay, so a vector is a mode of transportation or a kind of like a midway point. I'm trying to think of the best way to define it.
That has a lot of information in that one. Just looking at the picture itself, there's a lot of information there.
It's in the words, like the typed out part, it says it straight out. So what's the carrier? What's the host? Is another word for vector, if that helps.
That's it. So the plasmid and the vector.
Yep, the plasmid. And if you remember, the plasmid is just a circular piece of DNA that hangs out in bacteria along with their nucleus. So these are all their normal chromosomes over here. And a plasmid is just an extra piece of DNA. So the reason it's a vector or a carrier is because you can take that plasmid out and insert human genes into it. For instance, the gene for human insulin production. And once it's added in, you can put it right back into this bacterial cell and it'll replicate itself over and over and over again.
Pretty cool.
Yeah, it's way cool. Let me write this in for anybody else who's going to watch or see this later. But a vector equals a carrier or host. And plasmid is the circular DNA inside of bacteria. All right. During development from a zygote to a multicellular organism, the cells will specialize. This process is called
I have to find that.
Do you have a guess before you find it?
No.
What topic? It's in cloning. I believe it's in stem cell cloning. Yes.
Oh, during the development of a zygote to multicellular, thought I knew it.
Oh, there goes my watch. It's on slide 29, or in the textbook.
I didn't know if zygote is a stem cell.
A zygote. Yep, differentiation. You're right, Shivani. A zygote is a fertilized egg.
Oh.
So going from that one cell to a multicellular organism.
It's called differentiate.
Yep, differentiation. And that's important because that's how we get all of our different types of cells. Not every cell in our body is a skin cell. Not every cell is a muscle cell. We need all these different types, yet they all have the same genes. Every single cell has the same genes, but depending on which genes are available to be replicated, determines what kind of cell it'll become, what it'll differentiate to. All right, who can describe the gene cascade process? I'll give you a hint. It has to do with homeotic genes, which we went over last week.
Homeotic gene. What is gene expression? Homeotic genes, master switches. Start with the genes cascade.
Shivani looks like she might have gotten it. I see a light bulb O.
Is a gene cascade
Ask that one more time. You cut out, Brandon.
Is it with the cloning?
Not with cloning, but with everyday gene expression. So, Shivani has. Oh, go ahead.
So with the cascade, I got it. I think so. That's fine. And alright, it'll take a second.
No worries. Take your time.
Let's look technically. As our DNA goes throughout our body, it closes and opens for transcription. Is that what gene cascades is?
Similar. So
during the levels of packaging in our DNA, certain part of our DNA is open and the other part of it's closed.
Uh-huh.
Okay?
That is right.
So with our brain cells, it's open in one part, and when another part, that same part of our cell is going to be configured somewhere else in our body, not the same.
Yes.
Is that the answer right there?
Yeah, this is the answer right here. So you're right in your thinking, but gene cascades happen within a single cell. So, like Shivani wrote on the side, as one gene turns on, which it starts with a homeotic gene, because it's the master switch, right? So as the homeotic gene turns on, and it produces a product, that product goes down the line to another gene and causes it to turn on. And so it kind of starts a domino effect, I guess. And it has to do it in specific timing, or else the order gets mixed up and you get funky looking proteins that aren't folded correctly. So a homeotic gene gets turned on, and then it has a domino effect turning on other genes in turn along that open DNA section. Any other questions with homeotic genes or gene cascades?
No. Not here.
Okay. Any for you, Shivani? Awesome. So what's the purpose of PCR? This is in the biotechnology and gene expression. Another name for PCR, just a reminder, is polymerase chain reaction. The picture on the slide looks kind of like a bracket. Exactly, Shivani. It amplifies small amounts of DNA. So if you take a single gene and you copy it into two, then you can take each of those two and copy those till you get 4, and then 8 and 16, and so on and so forth. And that's